This is the second webinar on the MMRRC strain GQC report. Here, we address strain classification. Genetic complexity of a mouse strain is a fundamental characteristic with broad implications on colony maintenance, experimental design, and interpretation of results. In this video, we will briefly explain how genetic complexity is characterized and presented in the MMRRC strain GQC report. Once the combined mini-MMRRC report is generated, the strain is assigned to one of 7 possible types, based on genetic complexity. These types are based on standard classes recognized by nomenclature rules and used frequently in the literature. We have codified these strain types to reduce ambiguity, and we divided the congenic class into 2 types, congenics in the narrow sense, and incipient congenics more broadly. Let's review these strain types from the least to most complex. Co-isogenic strains are the simplest genetically, as the donor and recipient are the same strain or substrain. Only one genetic background is present in these strains. The names of these co-isogenic strains specify the genetic background, the locus and allele of interest, and the current holder. Congenic strains have genomes composed of 2 genetic backgrounds. The donor strain is found only in a single chromosome spanning the locus of interest. The recipient background is found everywhere else. The names of congenic strains begin with the recipient and donor genetic backgrounds, in that order, separated by a period, followed by the locus, allele of interest, and the current holder. Incipient congenic strains are also composed of 2 genetic backgrounds, but include some donor background contribution in 1 or more additional chromosomes. The names of incipient congenics follow the same rules as those of congenics. Note the use of the period to separate the recipient and donor background strains. The level of congenicity in incipient congenics may vary dramatically. For example, compare these 2 ideograms. Both are incipient congenic strains. Crosses are strains with non-replicable genomes derived from 2 inbred strains. The line separating crosses and incipient congenics has always been somewhat blurry, but our classifier uses the following rule. If a background contributes more than 80% homozygosity in the genome, the strain is classified as an incipient congenic. Otherwise, it is a cross. We acknowledge that this threshold remains arbitrary. Our selection of 80% homozygosity reflects the standard nomenclature requirement for at least 5 generations of backcrossing for a strain to be considered a congenic, and the stated goal of the MMRRC strain GQC report to provide potential users of a strain with actionable information for experimental design and result interpretation. If you are interested in learning more about the expectations for strain congenicity, please see the dedicated video. In addition to the locus, allele of interest, and holder, the names of crosses list 2 genetic backgrounds, separated by a semicolon instead of a period. Congenic, incipient congenic, and cross strains may include a limited contribution of a 3rd background strain. This contribution must be limited both in the fraction of the genome affected and in the number of locations where that 3rd background is found. The presence of a 3rd background is indicated in the strain name by adding a parenthetical identifier after the recipient and donor backgrounds. This is either the name of the 3rd background strain, if it is known, or CG, for complex genetics, if it is unknown. Stocks are strains with non-replicable genomes whose genetic backgrounds cannot be ascertained with the current mini-MMRRC pipeline. These strains are named stock plus the locus and allele of interest, or they have widely recognized names like Swiss Webster or CRL-CD1. The product of the MMRRC strain classifier, the strain type, is used in the summary section of the strain GQC report and must be consistent with the name shown in the report header. Importantly, the strain type also determines the report content. The ideogram, genome analysis, and estimated number of segregating SNPs are not shown in the MMRRC strain GQC report for crosses and stocks because their genomes are not replicable. These figures are available upon request, but they do not accurately represent the genome of the strain. The ideogram provides a simple way to confirm the strain type classification for co-isogenic, congenic, incipient congenic, recombinant inbred, and consomic strains. More details can be found in the following publications. Additional webinars will be created and posted on the MMRRC website. We would like to thank our funding sources and everyone involved in the MMRRC strain GQC project.